Cajuns are French. That's the story. French settlers arrived on the maritime coast of Canada in the early 1600s. By 1750, they'd grown from roughly 50 founding families into a population of over 10,000, all Catholic, all speaking the same dialect, all farming the same salt marshes along the Bay of Fundy. In 1755, the British deported them. Some ended up in Louisiana. They kept marrying each other, and their DNA is French, with some Native American on the side. That is what most people who've researched this topic already know. That story is not wrong. It's just the least interesting thing that's genetically true about the Cajun people. Baton Rouge, Louisiana, 2003. A researcher named Mark Batzer, George C. Kent Professor of Life Sciences at Louisiana State University, is looking at population frequency tables for a mutation called C.216GJ. He knows what an outlier looks like. What he's looking at is not an outlier in the normal sense. The number is so far beyond what his model predicts that he checks the sample. Then the methodology. The mutation is real, its concentration is real, and nobody can fully explain it. The Cajun population is one of fewer than a dozen populations in recorded human history where researchers can trace the precise moment a mutation entered a gene pool, watch it amplify across fully documented generations, identify living carriers today, and sequence the medical consequences in real time. This is not a heritage story with some interesting DNA at the end. It is a forensic case, and the evidence has been building inside a living community for 270 years. Stay with me, because this next part is the reason this video exists. If you've tested your DNA and your results show French ancestry, French Canadian or Quebecois roots anywhere in your family tree, what you're about to hear applies to you in ways no ancestry website will ever flag. Subscribe to Coded in Bone because this goes deeper than a heritage result. Dr. Branya Keats spent 30 years studying Acadian genetics at the LSU Health Sciences Center in New Orleans. In 2001, she organized a symposium called Genetics of the Acadian People at Nicholls State University in Thibodaux, the heart of Cajun country. She opened with a finding that has not been widely reported outside genetic circles. The Acadian population carries the genomic signature of not one bottleneck event, two, and the second one is still running. The entire field agreed this was a French story. The data says the most important ancestry in this story left no name, no record, and no recognition in any history book written about Louisiana. Before we go any further, drop in the comments. Where in the world do you trace your ancestry? I read them. Because by the end of this video, your answer may connect to what I'm about to show you in a way you're not expecting. No name. That is the precise problem. A Cajun man from Louisiana, surname French, Catholic, roots documented back 14 generations, submitted his Y-DNA for testing. 14 generations. Church records, land deeds, baptismal registers. His family had been French since before anyone in his family tree could remember being anything else. His result came back, haplogroup C/22870. That is not a French haplogroup. That is Mi'kmaq, the indigenous people of maritime Canada. 14 documented generations of French names, and underneath every single one of them, a paternal lineage the parish records never recorded. And yet, the data said the exact opposite of what the documentation showed. The genealogist said French, with a little Native American. But in a significant number of Cajun family lines, the Native American ancestry isn't a faint admixture signal in the autosomal data. It is the direct paternal line, the Y chromosome, the line that's hardest to obscure. And the reason it was obscured isn't complicated. Mi'kmaq men who married into Acadian families often received French surnames at baptism. They entered the record as French. The genome never agreed. Roberta Estes has been running the Acadian Amerindian Ancestry DNA Project for over 18 years, documented genealogical verification across Louisiana Cajun families tested against full mitochondrial sequences and Y-DNA haplogroup analysis. What it has confirmed is multiple distinct Native American lineages, both maternal and paternal, within what the official record describes as a wholly French-descended population, not one family, a structural pattern across the population. Françoise Godet was born in Acadia around 1621, mitochondrial haplogroup J1b2, European. Her direct matrilineal descendants have been confirmed through full sequence testing across multiple families now living in Louisiana. She is real. She is traceable. She is European. But she is not the only founding lineage, and the others have been far harder to find. Not because the DNA is absent, because the records were never designed to look for it. Colonial parish records are not a genetic database. They are a record of who was named, baptized, and counted, which in 17th century Nova Scotia meant French and Catholic. Anyone who converted, who took a French name at the church door, vanished from the documentation as anything other than Acadian. The genome didn't vanish. In some Cajun family lines with French surnames going back to the 1600s, lines with genealogical societies devoted to preserving them. The dominant paternal haplogroup is Mi'kmaq. No Mi'kmaq surnames, no Mi'kmaq records, no place in any Louisiana school's history curriculum, just the Y chromosome, still running. A surname is not a genome. Bay of Fundy, early 1600s. The first French settlers here are not soldiers. They are farmers, fishermen, engineers of a particular kind. People who look at a tidal flat and see farmland if you can keep the ocean out. They build dikes along the salt marshes, earthen banks, wooden sluices, tidal engineering that takes generations to get right. They don't figure it out alone. The Mi'kmaq people know this land. The children born from the exchange between those two worlds don't have a column in the census, but their genomes are still here. Simon Gravel and his team at McGill University published a study in 2013, peer-reviewed, in PLOS 1, examining the founding genetic pool of the Quebec population, which shares its ancestral base with the Acadians. They found consistent Native American admixture at approximately 1% genome-wide. In individuals whose genealogical records showed 0, Native American ancestry, 0 on paper, there in the DNA, consistent, genome-wide. But here's where it gets strange. A 1% admixture signal spread across 500,000 people is not a footnote. Genetically, it is the difference between a disease mapping study that produces accurate results and one that generates false negatives for an entire ethnic population. It had been assumed away for decades. Building on that work, a Montreal Children's Hospital team, Eberman, Kanekoop, and colleagues traced the USH1C mutation to the shared founding pool of both the Quebecois and Acadian populations, not arrivals after 1650, before. The mutation was in the gene pool at the moment of foundation, before the community had a name, before a single French child was baptized in a maritime Canadian church. The population the British deported in 1755 was not genetically what it was documented to be. Older, more mixed, more genomically complex than any census or parish record reflected. The deportation didn't just scatter French Catholics. It scattered a living genetic archive and then closed the lid. That mutation had already survived the founding bottleneck. 50 families becoming the genetic ceiling for 10,000 people. Then the deportation. Then Louisiana. 3 compressions across 150 years. Each one drove the allele frequency higher, and the descendants carrying it today have, in most cases, no idea it's there. Carl Heck had no idea. Southwest Louisiana, sometime in the 1990s. Carl is not a geneticist. He's a man researching his family tree, the way a lot of Cajun families do, because the records are good and the community is tight-knit and the surnames go back a long way. His mother was born Verna May Talbot. The Talbots are one of the original Acadian families in Louisiana, one of the lines that came over from Nova Scotia after the deportation and stayed in the bayous. Carl isn't looking for a diagnosis. He's looking for his people, but he keeps noticing something. The same condition, Usher syndrome, hereditary deaf blindness, keeps appearing across families in his community. Too many times for coincidence. He finds a flyer for a symposium at Nicholls State University in Thibodaux. He drives out. He brings his son. Usher syndrome type 1C causes 2 things. First, profound congenital deafness. Children are born unable to hear. Second, progressive vision loss beginning in adolescence, leading to full blindness by early adulthood. In the general American population, USH1C accounts for about 2.5% of all Usher syndrome cases. In Southwest Louisiana, among people of Acadian descent, a single mutation, C.216G in the UH1C gene, accounts for virtually all of them. Southwest Louisiana has the highest concentration of deafblind citizens in the United States, not one of the highest. The highest. This isn't just ancient history. This is in your DNA right now if your family came through this population. 2 carriers of C.216GJ who have a child together give that child a 25% chance of being born deaf and losing their sight before they're 30. In a community that has married within itself for 270 years, the probability of 2 carriers finding each other is not a statistical edge case. It is a built-in feature of the population. Carl Heck walked into that auditorium not knowing that what Dr. Branya Keats was describing was in his family. He didn't know, before the day ended, that the Talbot line he'd spent years tracing with such care carried a genomic inheritance that predated Louisiana entirely, predated the deportation, predated any chapter of American history his family had ever discussed. He learned it from a geneticist. On a Tuesday, the Talbot connection to USH1C was confirmed. The mutation had entered the gene pool before 1755, before his ancestors were put on a British deportation ship, before they found the bayous and stayed. His family had been carrying this, silently, in exactly the way genetics predicts a recessive allele moves through a closed population, since before the British government decided their land was worth more than their presence. In our last episode, we tracked how the Ashkenazi Jewish founder effect concentrated both extraordinary cultural continuity and elevated disease risk within a population over centuries. Right now, we're watching the same mechanism, but this time the bottleneck wasn't ancient. It was created deliberately by a government in 1755. Drop a comment if you see the parallel. The science here is even more precise. The mutation doesn't cause any symptoms if you carry one copy. Invisible, silent, no sign. For 270 years, Cajun families passed it to their children without knowing, and without any way of knowing, until a genetics lab developed a test for it. Testing changed everything. By 2003, Mark Batzer's lab at LSU, working alongside Dr. Mary K. Pelias and Dr. Bronya Keats, had traced the mutation, identified the gene, developed a carrier test, and established exactly when, in documented history, the allele entered the Acadian gene pool. The science was finished. The problem was not. The USH1C gene normally produces a protein called harmonin, critical infrastructure for both the inner ear hair cells that process sound and retinal photoreceptors that process light. The C.216G mutation introduces a defective splice site that causes the gene to produce a truncated, non-functional version of harmonin instead. No functioning harmonin in the inner ear, deaf at birth. No functioning harmonin in the retina. Vision loss through adolescence, both at once, one letter, G to A. What they found next changed everything they thought they knew about the scope of the problem. A gene therapy approach targeting exactly this splice site is being developed, and in principle, it could restore gene function, prevent the deafness, stop the vision loss in children with 2 copies of the Acadian allele. But gene therapy requires early diagnosis. Early diagnosis requires testing before symptoms. And testing before symptoms requires a community to know, at population scale, that this mutation is in them. The same year, a team at the Université de Moncton and Horizon Health Network in New Brunswick published the first large-scale formal carrier screening of the Acadian population as a distinct genetic group. What they found confirmed what Louisiana data had suggested but never formally established. The Acadian population carries multiple founder effect alleles at elevated frequencies, not just USH1C, tyrosinemia, Fanconi anemia, NPHP3, nephronophthisis. The list kept growing. The bottleneck didn't concentrate one allele. It concentrated everything that happened to be in those 50 families when the founding event closed the genetic pool. That mutation, C.216GJ, isn't only a medical finding. Through haplotype analysis, researchers can trace it to specific ancestral founders alive in Acadia before 1650. It is a genomic timestamp, a molecular record of events from 400 years ago. It is still there. It is still speaking. Some argue it was never as loud as I've made it sound. Here is the strongest possible case against everything I've shown you. Argument 1. The Cajun population has been studied specifically because of Usher syndrome, not the other way around. Every isolated population carries elevated disease rates. What makes the Acadians look exceptional is that they have a state-funded genetics program watching them. The data may be real. The story built around it may be a research artifact. Argument 2. The USH1C founder mutation isn't unique to Louisiana. Quebec carries the same allele. France itself does not show elevated rates. If the allele was already amplified before Louisiana, what exactly did the Bayou add? Argument 3. The Native American admixture in Cajun DNA is roughly 1% genome-wide, near the noise floor of routine ancestry analysis. Calling Mi'kmaq ancestry a structural feature of the Cajun genome may be overstating what the data shows. And yet, the data said the exact opposite of what those arguments predict. The New Brunswick study, published in 2022 by Langlois and colleagues, was conducted on a completely separate Acadian population, one that never passed through Louisiana, never settled the bayous, never came within 1,000 miles of the Gulf of Mexico. That population showed the same elevated carrier frequencies for USH1C and multiple other Acadian founder alleles, independent of Louisiana, independent of LSU's research program, independent of any reporting bias argument tied to a specific university or clinic. The counter-argument was good. It wasn't good enough. What the New Brunswick data proves is that the amplification happened in Acadia, in the founding event itself before the deportation. Louisiana preserved what was already concentrated. New Brunswick preserved the same thing, separately, in parallel 2,000 kilometers away. Same genome, different continent, same result. And the USH1C mutation traces, through haplotype analysis, to founders who arrived before 1650. That is 105 years before the Great Deportation. The British didn't create this population's genomic singularity. They handed it to the Louisiana Bayou. Before the deportation, let that date sit. Grand Pré, Nova Scotia, September 5, 1755. British Colonel John Winslow locks 418 Acadian men and boys inside the Church of St. Charles des Mines. Outside, their families wait. He reads the deportation order aloud. 10,000 people, the descendants of those 50 founding families, carrying a mutation that has been amplifying in this closed gene pool for 150 years, are about to be scattered across a continent. Roughly half of them will not survive. Historical estimates place the death toll during the deportation period at close to 50%. Disease on the transport ships. Starvation in places that didn't want them. Some were sent to Virginia and never let off the ships, sitting in harbor for months before being redirected to England. The survivors who reached Louisiana settled along the bayous. Bayou Lafourche, Bayou Teche, Bayou Vermilion, far from New Orleans, far from the demographic mixing that cities produce. For the next 200 years, they married each other. The child being tested for US H1C at a New Orleans pediatric genetics clinic in 2024 carries a mutation that was already present in this gene pool in 1640. Colonel Winslow did not create that mutation. He ensured it had nowhere to dilute. A single British colonel, locking 418 Acadian men inside a church in 1755, accidentally created the most genetically studied founder population in North American history, and the mutation he concentrated is now blinding and deafening children in Louisiana at a rate that has no parallel anywhere else on this continent. Not because of poverty, not because of any environmental factor, because of a single nucleotide change, G to A, at position 216 of the USH1C gene, present in this community since before the Mayflower, amplified twice by historical violence, and still running forward through every generation born in Cajun country. The deportation is not history. In the genome, the deportation is a diagnostic finding. 1640 and 2024 are the same moment in the DNA of a child who is alive right now. A gene therapy targeting this exact splice site is in development. In animal models, it shows real promise. Early human work is underway. But for it to work, a child needs to be diagnosed before the hearing loss begins, ideally through newborn screening. And for that to happen, the entire Cajun community would need to participate in a population-level carrier screening program. As of now, that program does not exist in Louisiana. The missing variable is not science. Louisiana has known about this for 30 years. The Center for Acadiana Genetics and Hereditary Healthcare has existed within the LSU Health Sciences Center for decades. The carrier test has been available there for over 20 years. Dr. Keats held her symposium in 2001. Carl Heck drove to Thibodaux and found out what his family was carrying. The science is not missing. It has been sitting in a New Orleans clinic, available to anyone who knows to ask, for 2 decades, and yet, as of 2022, no population-level carrier screening program existed in Louisiana targeting Acadian descendants. The New Brunswick study, the first formal large-scale screening of the Acadian population as a distinct genetic group, was framed explicitly as addressing a community that had never been investigated at this scale, not investigated. In 2022, for a mutation Louisiana researchers identified in the 1980s, there are currently children in Louisiana who have been born with 2 copies of C216GJ. They are not yet deaf. Their hearing loss will begin in infancy. Their vision loss will begin in adolescence. The gene therapy targeting their exact mutation is in development. The window for early intervention, the window where treatment could prevent what is coming, is measured in months from birth. Dan and Annie are a married couple of Acadian ancestry in Southwest Louisiana, both carriers, both documented in a case study used in university genetics education. Their community is the largest population of deaf-blind citizens in the United States, living in the same parishes, worshiping in the same churches, carrying the same mutation that crossed the Atlantic on a deportation ship, waiting for a health infrastructure that has their genetic profile on file and has not yet shown up at their door. Knowing about a genetic risk and acting on it at population scale are not the same problem. The first was solved in 2003. The second has not been solved. The gap between them is not scientific. The British deportation of 1755 ended as a geopolitical event in 1763 with the Treaty of Paris. As a genomic event, it has never ended. The same fault line, a small isolated population, a concentrated mutation, an absent infrastructure, runs in an unbroken line from the Church of St. Charles des Mines to a pediatric genetics clinic in Lafayette. Same mutation, same fault line, same absence. Nothing ends. That is what the genome keeps saying. The 50 French families who arrived at the Bay of Fundy before 1650 are still here. Not in any church record or genealogy database, in the US H1C allele frequency tables, in the haplotype blocks, in the mitochondrial haplogroups of people in Thibodaux and Lafayette, New Orleans, who have never thought of themselves as a founder population. They think of themselves as Cajun, Catholic, French. The genome holds a more complicated story. If your ancestry includes French, French-Canadian, or Quebecois lineage anywhere in your family tree, the probability you carry C216G is not zero. The carrier frequency among Acadian descendants has been estimated at between 1 in 20 and 1 in 30. That is not a rare disease carrier frequency. That is a common allele in a specific population. And that population is not only in Louisiana, it is in New Brunswick, in Quebec, in Nova Scotia, in every harbor where a deportation ship made land and the survivors stayed. If your family came through that diaspora, this may have come with them, not as a burden you choose to carry, as a fact that exists whether you know about it or not. Everything I just told you, hold on to it, because it means something different now than it did when this video started. This has not been a story about Cajun people as victims. It has been a story about what history does to DNA, and what DNA does with history. Those are not separate processes. They never were. The migration and the mutation are the same event, viewed from different angles. If this reached something in you, subscribe to Coded in Bone, because the next episode goes somewhere the textbooks never thought to look, and you'll want to be there when the data arrives. In 2003, Queen Elizabeth II issued a royal proclamation apologizing for the forced deportation of the Acadian people. No proclamation has ever been issued for what the deportation did to their genome, and no proclamation would change it.